Welcome, everyone. Thanks for coming. I'm sure you all know this is Curiosity Quest Night, and we'll have 5 different presentations tonight. And the first presentation is going to be from Azir and Will. They're going to talk a little bit about what happens when you take in food. We're going to start in about 30 seconds. I wanted to just begin by saying that I love this night, and I want to thank Dr. O'Connor for sort of coming up with this whole event and this whole idea of having Curiosity Quest. It's been going now, I think 4 years? 4 years. So this is our 4th year of doing this, and among the many things that I love about it, one of my very favorite things is just how supportive the audience always is of their friends who are presenting. So I look forward to that same energy that we have every year. And I've actually already seen it as the presenters were warming up, the members of the audience were cheering them on. And so thank you for that. And I'm going to ask Will and Azir to come on up. And I promise you that there's a laser pointer on its way. It will be here before you get to a slide that needs a laser pointer, I'm hoping. Okay? All right, but we should get started. So come on up. We're going to just see if we can get 2 microphones. Second microphone.
It's the computer right there. It's the computer right there on the thing.
Is this working?
Oh, okay, yeah.
Is it working?
Yep.
Do you know how to airplay?
Yeah.
Are you sure?
You guys are confusing. Wait, this is not ours.
There you go. Yep. There you go.
Okay, good evening, everyone. My name is Azir.
I'm Will.
And glucose to beats per minute. Laser pointer.
So my question was, like, how does heart rate, like your beats per minute, affect how fast blood or glucose can absorb into your bloodstream after a meal? In this case, would be apple juice. So let's find out.
Okay, our curiosity. So the circulatory system and the digestive system are connected, and we say this because when you consume food, it goes through your digestive system, and the digestive system transfers the nutrients, in this case glucose, which we're focusing on today, into the circulatory system. And so we hypothesize that the speed at which sugar can be absorbed into the bloodstream is dependent on 2 factors, and those 2 factors are the speed at which your blood is pumped, so that your beat per minute, and the speed at which you can digest food. And so we decided to study the first one, how fast your blood is being pumped throughout the body and transferring that glucose to different parts of your body that needs it. Okay, our experiment.
So the experiment, it has a couple of things that you need to do. One thing is fasting for 5 hours prior to the experiment at least. We'll talk about that later. And so we test our glucose 5 minutes before we drink the apple juice to get like a baseline. And then we test it immediately. And then we test it every 5 minutes prior to drinking the apple juice. And yeah, that was how we got our data.
And we do this 5 minutes for 60 minutes.
And also with the heart rate, we would just take our heart rate after every measurement, and then at the end we averaged it and got the averages that we'll be using for like resting and high exercise. So.
Yeah, we took our heart rate, if you guys are wondering, with this watch right here.
Yeah.
Okay, so our independent variables. And so what we are changing about the experiment is how our beats per minute, essentially. And so we're exercising and then we're resting. And so exercising, we're getting our heart rate high so that the blood is moving faster throughout our body, and we're resting so that it's moving slow.
And just to clarify, like, when we were exercising, it was pretty vigorous, like running, like standing, jumping up on tables and chairs. It was pretty wild, you should have been there. And resting was just sitting down, like doing not much. And yeah, that's it.
Yeah. Anything at all, just sitting down, pretty lazy actually. So our confounding variables, and so Will mentioned fasting earlier, and so fasting is important because in order to have a good experiment, you need to be starting with a low glucose, which is milligrams per deciliter, and you need to have a low one because if it's high and you've eaten prior to the experiment, your glucose levels won't be just based on the apple juice that you're consuming. The food you ate before that will also affect the readings that you get. And so you need to fast in order to have a good experiment. And we know this because we actually have one failed experiment that we don't show you guys today because of that specific reason. I ate before the presentation. I meant the experiment and I messed it up, but yeah.
It's fine, we get hungry. So the results, this is what we've been waiting for. You see the one on the left is no exercise, and the one on the right is with exercise. And the things to notice are with exercise, it rises like a lot higher, like the peak is much higher. That's not as important as the fact that it rises quicker. And you'll be able to see that more clearly when we overlay the graphs. But yeah, that's pretty much it for just like the two graphs by themselves.
All right. And so this is the two graphs that you guys seen combined together. And so once again, just to go back to make it more understanding. Yeah, so we have a blue and a red one. It's the same blue and red lines here. Okay, our observation. Like he said, the glucose levels are rising sooner, and our likely reasoning, our blood is pumping faster. Therefore, our bloodstream must be absorbing the glucose at a much faster rate. You can see this, show this section. Right.
Is it, but it's not working, but it's alright. So you can see that like all the points where it rises are a little bit on the red line are a little bit farther to the left than they are on the right line. And that pretty much what it's showing is that it rises sooner after drinking the apple juice when your heart rate is higher. And so we were really excited to see that because it seems to like support our thesis, our hypothesis. And you want to talk about the error bars?
Yeah, okay, so the bars that you see, like surrounding the points, I guess, are called error bars. And we got these error bars by using standard deviation. And so standard deviation is the possibility of where that point could have landed. And so it could have been, like, for example, one of those points could have been anywhere along that line. Yeah, and so like, it's an average of all of them, and that's like a possibility of where it could have landed, but that's where it ended up.
Another thing that we didn't mention really was that these lines like aren't our only experiments. We did like numerous experiments and we had to average all the measurements. So this is like, I think these lines are like 5 experiments condensed down into 2 lines. And that's how we got the standard deviation because we had to like compare it to a lot of different experiments, so yeah. Yeah,
just to give you guys a better understanding, you see where on the x-axis time in minutes, it says 0. There's this big arrow saying apple juice. This is the exact point in time where we consumed the apple juice. And then throughout, we were exercising or resting, and we were testing it every 5 minutes. And every 5 minutes, we would prick ourselves using a prick, or I don't know the actual device. And then we took the glucometer, and the glucometer basically tastes our blood to show how sweet it is in simpler terms, and it gives us milligrams per deciliter in our blood.
So what does this data suggests, pretty much what this data is suggesting is that the higher your heart rate is, the quicker your blood glucose will rise. And this is like what our whole experiment was centered around. So it was really cool to see. And also another minor thing is like after the initial spike, here if we go back, after the initial spike, which is around the whole working, you see like where it's 152, it goes down and then it spiked even higher. So that was really cool because unlike the resting where it kind of spikes at a really clear point and then dies down, it kind of spikes up and then keeps like going down and spiking. And that was really cool to see. So.
Yeah, and just to like oppose our observations and conclusion, as I explained before about the digestive system and this like circulatory system, apple juice, there isn't glucose in apple juice, and when you consume apple juice, you have to use enzymatic processes within the digestive system to break it down, and that glucose is transferred to the circulatory system. And so these observations just could be a coincidence, and there certainly are other factors that go into how fast the glucose is transferred into our blood because of these enzymatic processes that actually break it down into glucose. So is there correlation? The truth is, we're not really sure. The data is very clear and it suggests what we hypothesize, but yeah.
Thanks, guys.
Oh, sorry.
Just a finding word, I'm sorry. What we could have done differently, like obviously we had a lot of time, but we didn't have like unlimited time. So some things that we would have done differently if given more time would just like get a higher quantity of data, because we did collect like a good amount of data, but it couldn't hurt to have more.
Yeah, just to mention, we were pricking ourselves 20 times per class.
Yeah.
And we had about a lot of classes. So yeah. And the experiment we envisioned was a bit different from the one we have presented to you guys today, because we were going to take our blood throughout the day, not correlating to our class schedule times. And also, if we had more money, we would purchase these devices that would track our glucose throughout the day without us having to prick ourselves as many times as we did, which would have been much easier. And we would, like Will said, perform our experiments on other people to have a higher quantity of data. And that's it. Thank you, guys.
Thank you.
It doesn't really hurt me, but does it hurt Will?
It was excruciating.
Amanda? Oh.
So we talked about this to Mr. O'Connell a lot, and one thing we didn't really go too in depth in about like in here was something called the Glycemic Index. So like a lot of foods and drinks in this case are given like a score that's called a Glycemic Index, and it's really, for apple juice, it's really high, and it pretty much just says it has a really high sugar content, and your body's able to digest it and then process it really quickly. So since we were doing our experiment in like only an hour time frame, we used apple juice because it acts like really fast.
Yeah, it was the most convenient. The glycemic index, like he said in simpler terms, is how fast the glucose is coming into your body. Luke.
How much apple juice did you drink?
So, the graph that we showed you in the zero, like when we drank the apple juice, we were drinking approximately, well no, exactly 350 milliliters of apple juice each time. Zach, in the back.
So could you go back to the slide where you had both of them, both of the graphs each other? Yeah. So I had a question about the descent for the blue seems to be at a much slower rate than the descent of the red. What do you make of that?
Ooh, okay. Give us a few moments, Zach.
I mean, this is one of the things that we were kind of unsure of because in our experiment, we were really just studying like how quickly the glucose is gonna spike and how quickly it enters like the bloodstream. And we weren't really concerned with much that was happening after that. So I think we need a little bit more data and we need to focus it in a little bit better and a little bit more on like what happens after the spike. Like to answer your question properly, but
Yeah.
I'm sure.
Yeah, and like we said, like the digestive system and the circulatory system, there are a lot of things that we haven't researched, and there are a lot of things, a part of this process that we really don't know about. So it's kind of hard to explain the aftermath of what we're actually examining, which is the start, the rise of these two lines.
So at the start of your presentation, you said that there were two variables that affected the data you thought you were going to get. Did you try to control the other variable, or did you just only look at the?
What other variable are you referring
On
to?
your first,
Well, we were just pretty much saying that like we were gonna study this variable. And the other variable, like the enzymatic processes and stuff, like that is another something that could provide like almost a wall to the speed, like how fast it can go, but we didn't go too in depth about that. And yeah, that would have been like a totally different experiment.
Yeah, and we actually had another variable, which was medium exercise, I guess, which was just us walking around, and like a person would throughout the day. But we chose not to show you guys because, yeah, we didn't get enough data, and it didn't, like there weren't enough drastic changes between the two, like there were between high exercise and low exercise, or no exercise. So yeah.
All right, thanks very much. Thank you. Next up is Emma and Eleni.
Okay, hi guys. So, my partner's not here right now, but this is Eleni and I's curiosity quest. Like many of you, our project started by walking around in the woods and kind of looking for something that we thought would be interesting. And one of the observations that we made were all of those things. They were kind of like butt-shaped and all over the ground. If you go now, you're not gonna see them, and I'll explain why. But they were everywhere, and we were kind of wondering what they were. We recorded a lot of observations on them. This is just like one example of one skunk cabbage that we looked at, and we recorded them all in my notebook and looked for it, and then when we went back to Dr. O'Connor's classroom. We researched it to try and figure out what they were, and I'm not going to try and pronounce the Latin, but they're called skunk cabbages. So they have a lot of interesting things about them. One of them being that they're called skunk cabbages because they smell really bad, and I can confirm from personal experience, they do smell really bad. The other interesting thing about them is this really big word called thermogenesis, which they do, and it's super important to the skunk cabbage as a plant, because they come up basically before everything else. And what thermogenesis does is it warms the skunk cabbage so it is able to break through frost and snow before all the other plants. And you guys are all familiar, but I'm just gonna refresh what cellular respiration is. It's like taking in glucose and oxygen. For us, that would be eating food and breathing so that we get carbon dioxide. Well, and then the byproducts being carbon dioxide, us exhaling, water, the water vapor, and ATP, which is energy. One thing that I feel like a lot of the times people forget is that people use cellular respiration. Plants also use cellular respiration in addition to photosynthesis, and another byproduct is that it produces heat. And thermogenesis. That is a kind of cellular respiration. For the skunk cabbages, it happens in the roots of it, and essentially the more oxygen that they take in, the more heat that they're able to produce. So we wanted to study this more, and particularly study how much heat a skunk cabbage is able to give off, and we were going to do this by measuring the soil temperature. Our question became, how do skunk cabbages of different sizes affect the amount of heat that they emit? And one of the assumption that we kind of made, well, the bigger the skunk cabbage, the more heat that it's gonna give off. And this was our hypothesis. So how did we collect our data? Well, first we used a faulty thermometer, and then we got a better one. So our first day of data was not really reliable, but essentially we made a gradient. And what this means is, using the working thermometer, we would put it in the base of the soil, like right at the base of the skunk cabbage, and test that. And then at 1 inch increments, we measured the temperature going outwards. And this is kind of what that would look like, drawing courtesy of Eleni. We measured a lot of different factors, like leaf number, height, leaf width, circumference, and soil temperature at all these different places. We didn't use all of that data just because we kind of measured different things on different days, and eventually we came to height and leaf width being the two factors that we wanted to focus more on. This is us measuring. And this is all of the data that we came up with. Well, not all of the data, we measured other factors. This is the data that we actually graph. The red is because we didn't have leaf width for those days. And one of the things that we were wondering is, how do you show this gradient? So this is the temperature gradient, and we averaged out all of the numbers on the farther side of the graph. We measured, we averaged the base numbers, then we averaged all of the 1 inch numbers, and so on. And then that gets us to this gradient, which clearly shows that closer to the skunk cabbage, it is warmer, and as you get farther away, it cools down, and this kind of proves that the skunk cabbage is giving off heat. And this is our first graph, comparing it with another factor. And I want to explain what our x and y axis is. So on the y axis, we have a change in temperature. And as we were wondering, how do we show this data, we knew that we couldn't show every temperature from every point. So what we did is we subtracted our 6 inch numbers to the base numbers. And this tells us how much the temperature has changed. The more the temperature has changed, our idea was the more heat the skunk cabbage is giving off and the more energy that it's expending. This data is inconclusive because 0.057 is a really low R² number. So fortunately for us, we have this graph. And this graph is saying that the smaller a skunk cabbage is, the more the difference in temperature it has. Well, what does this mean? This is different than what our hypothesis originally was, because as you can see in this graph, all of the bigger skunk cabbages, which are going to be on that side of the graph, have a lot lower change in temperature. And as we were kind of discussing it, we realized this actually makes sense, because the bigger a skunk cabbage is, the outside leaves, when it's kind of poking through, are called the spathe. The bigger the skunk cabbage is, probably the easier time it's going to have breaking through the snow. So it does make sense that the smaller skunk cabbages would produce more heat, because they're going to need the heat more than the bigger skunk cabbage. And now, when you walk in the woods, you're not gonna see the pointy skunk cabbages that are there, but you're gonna see a bunch of leafy things that are a lot bigger than that. I actually just was out there today, and they're like giant, giant lettuces. So what are the strengths of our project? Well, first we collected lots of different variables of data, even though we didn't graph them all, because we were not gonna make an assumption about what could impact the heat of the skunk cabbage. After our faulty measuring tool, we used a working one, so our data was precise. We took lots of pictures, and we were using precise units of measurement. Now, the weaknesses are really important to look at because this kind of shows how reliable our data is. One of the important things to think about is that what was in the middle of our curiosity quest? Spring break. And a lot can happen over spring break. For example, we saw those before leaving, and then afterwards, giant cabbages. So our data there could have looked a little different. It's also a lot warmer outside after spring break, and it's just, that was an inconsistency. So I'll discuss what we would have wanted to do. Time, again, spring break, we didn't, they've grown over time, so that could impact it. And sunlight distribution and disruption. The sun that's shining on soil is gonna be naturally warmer than the soil that isn't being shined on by the sun. So that was something that we didn't consider while we were taking in our data. So our next steps would definitely be to get more data because, as Dr. O'Connor reminds us, you can never have enough of it. So we probably would want to get more data, and if we could go back in time, I would have wanted to get data from before spring break, more than what we had. We would also want a controlled environment, or maybe test for other things like sunlight and temperature. And speaking to having a controlled environment, we didn't test the temperature of soil that didn't have skunk cabbages by it, so it really would have helped in understanding that graph of the gradient, because it would have kind of shown how much the skunk cabbage impacts the soil more so. And also that they're in a lot of clusters, and that was one of the things that we took notes on in the beginning. And if we're measuring for one skunk cabbage, another skunk cabbage could be interfering with it if they were close enough. So just kind of controlling the environment. And looking forward, it'd be super interesting to look at the skunk cabbage on a molecular level and putting leaves of it underneath a microscope. That would just be cool to see. Yes, that's the end. Do you guys have any questions?
So did you ever do any tests on the soil when it was a leafy cabbage looking thing like you're talking about, or did you only ever do it on the spiky bugs?
Yes, so all of the data that is below the red chunk, that is all while it was super leafy, and that was when we're able to test leaf width also. And there is a difference in the numbers, obviously, because the bud is going to be smaller than the leafy thing. Dr. Hyde?
Thank you, Nathan. I was wondering if you could talk us through a little bit about why you chose to measure the temperature gradient rather than just the absolute temperature nearest to the skunk cabbage.
So I think that the more heat that the skunk cabbage is producing, the farther it's gonna reach. And it also, by testing a gradient, it means it's less impacted by the temperature outside. For example, if I take the temperature when it is freezing and I'm wearing 2 jackets and I'm still cold, that soil is naturally going to be colder than when it's a sunny day right from coming home from spring break. So it kind of helps show the difference in temperature and the impact that it's making, and also makes it more reliable.
Was the heat gradient you took, was that linear, or did you sort of try to do a circle around the whole plant?
We didn't do a circle around the whole plant. We would first put out our measuring tape and then just measure it like farther and farther away from it. As you know.
I've already said this, but like, did you ever test the internal temperature of the plant itself?
No, we did not, which would be interesting to see because in our research, we did come across the fact that there's like this tiny, I think it's what holds all the pollen, and that's what attracts bugs, and that's what smells bad, and that's like in the center of it. But also where thermogenesis happens is in the roots of the skunk cabbage. So we were pretty close because we like put the thermometer like right next to it, like we would like really put it as close as we could to the actual plant. Ted? Probably like this deep. Peter?
You said that thermogenesis, you think it occurs by cellular respiration. So, do you think other plants do this, or do you think it's the speed at which skunk cabbage does this that generates that much heat?
Well, one of the things that I would want to look into more is how much oxygen it takes in, because the more oxygen it takes in, the warmer it is. Thermogenesis is not unique to skunk cabbage. In fact, I'm pretty sure it is happening inside of us too, in like certain bones or something like that. But yes, the skunk cabbage does it more, and like I said, characteristically, like that's a big part of the skunk cabbage. Fun fact, it actually uses as much energy as a mammal of its size. So think of like a chipmunk that's the size of that, that's like running up and down trees and stuff. The stationary plant is using just as much energy, just to provide some perspective.
Gavin and Finn are next.
Bones. There we are. Sorry. Sorry. Bones. So, some background. We were walking through the woods, probably October. It was close to the beginning of the year. We weren't even thinking about the curiosity quest at that time. And we found some bones just lying in the grass, in the leaves. At first, we thought this was a skull. It's not. Gray told us it wasn't. But our starting questions, we had a few starting questions, which were like, what animal is this? And how did it die? And those could have been good curiosity quests. The animal was a deer, we found out pretty quickly. And we also found out that we did not have a full set of bones, which was a bit of a letdown. But so some important points of data to keep in mind as I take you through the presentation. Deer can live up to 15 to 20 years, and they mature from 3.5 to 4.5 years. So right off the bat, we needed to know what type of deer it was to collect data on the lengths and the size of the bones. And so the two most common deer in Delaware are white-tailed deer and sika deer, and I'm sure all of you are very familiar with white-tailed deer because they have a white tail and you see them everywhere. And there are 45,000 white-tailed deer in the state of Delaware as opposed to only 400-ish sika deer in the state of Delaware, and there are only 12,000 in the US. And so from this point forward, we assumed that it was a white-tailed deer and collected data as if it was a white-tailed deer, as the numbers pointed to it being one. So our question, the question that would cover most of our interests was, how old is this deer and what sex was it? And we were pretty confident that we could figure out quite a bit of information from this. So at the start, we thought that smaller bones would help us the most, and specifically the jawbone, as they would provide more accurate measurements, and if they're more frail, they would show some breaks and maybe give us clues to how they died. But we progressed, and we found that the femur and the pelvis would also give us very accurate measurements, and the pelvis especially determining the sex of the deer. So from that point forward, our three main focuses were the pelvis, the jawbone, and the femur. So for the femur length, we only had one femur, but we were very sure that it's a femur, even though there are quite a few tibias and fibulas down there. So on this graph, just focus on the blue graph. The green one is human, the other ones are pig and sheep and something else. But we're focusing on the blue graph alone. So our first measurement was the length, the total length of the femur. And we got 270 millimeters as opposed to the average, which was 280 millimeters. So these next couple slides are going to be quick, just measurements of the femur. So the intercondylar width, which is right between here, we had 11, and the average was around 12 to 13. The LAP length was 29 as opposed to the average being 33 millimeters. The ICH was 11 millimeters as opposed to the average of 13 millimeters. The MAP length was 24 millimeters as opposed to the average of 26. And the LML length was 23 as opposed to 25, and MML was 20 as opposed to 22. Now we have the big cross section of the joint, which we got at a 55 as opposed to 59 being the average for a femur. So here's all our data. I'm not sure if you can see them very well, but we have little green dots where our data points were, and you can see they're all just a little bit below the standard deviation bar for the deer. So the data we collected on the femur was in a study of around 16 deer, or a study of 16 deer, 10 of which were female, 6 of which were male, and they were from the ages 1 to 4. And so the femur pointed us in the direction of quite a young deer, maybe 2 or 3 or 1, because it was it was below average in almost every category. And we had to keep in mind that if it was a male deer, it would be slightly larger at a younger age because they develop faster, or they develop bigger. And now for our pelvis. The pelvis was a very vital bone to determine the sex of the deer. And we started off with a pelvic girdle, and I'm not sure if you can really see them quite well, but they're right there. And there are female pelvic girdles are known to be very small and triangular, and male pelvic girdles are known to be more ovular and large, and so ours are ovular and quite large. And so that points us in the direction of it being a male deer. And you can also see it's quite a narrow pelvis, which also indicates that it's a male deer. And another point of evidence is that it must have been, it points to it being a little bit of an older deer, as there's not a lot of cartilage that could have been in here that to decompose, as the younger deer have more cartilage than the older deer. It seems like a more developed joint. Now for the jawbone. The jawbone can tell us a lot about age, especially not much about sex, but right off the bat, when we found the bones, we knew it was an herbivore because of its more flat teeth. And upon further research, we were able to determine that it was an older deer off of this because the molars are further away from the jaw and because of how worn down the teeth are, which you might not be able to see. And the enamels were also quite worn, which indicated an older deer. And so through our research, the data we gathered from the jaw indicated that it would be around 5.5 years old. So here's the picture. That hole up on the, that hole up on the top right, that's not supposed to be there. That's probably some break or maybe been worn down since its death. So obviously we have some conflicting data because the femur is quite young and the jawbone says it's quite old. So one of the ideas that we had was maybe the deer had a lack of nutrition from a younger age. It wasn't able to develop fully, and therefore at 5.5 years old still had a small femur. Or maybe we got bones from 2 different deer. Maybe they were in the same spot because a fox would drag them there to feed its cubs or something. And so those were 2 possible options. Ultimately, our conclusion, we have 3 things that we were concluding. We had the safe assumption that it was a white-tailed deer. We had sufficient data to infer that these bones belong to a male deer, and the data points the age of the deer was around 3 to 5.5 years old if we take into consideration all of the bones. There's a little bibliography and questions. Max?
Where do you get your bones?
Well, so you know where those little trees are? Sorry. I mean the New Forest area-ish. Sorry. If you follow to the left of that and head down for a while and I'll show you if you want to see, but I don't know really how to describe it.
It was all in the same spot?
Yeah, it was all in a pretty close radius. Gwen?
Do you ever look at graphs for like the size, like the average femur sizes for like a young deer and compare it to those?
That was the one that we were looking at. We were looking at yeah, so oops, what? Sorry. We collected data from a website, the National Health Institute, something, that was the study of 16 deer between the age of 1 to 4 years old, which were considered young, considering that they could live up to 20. Eli? No, we found a lot. We found a lot of vertebrae. We found a lot of ribs. We found a lot of bones, but we didn't find them all. But we couldn't gather as much data from the other bones as these, and I probably wouldn't want to bring them all over here. Lindy?
Do you think you could clarify some of your data that you found inconclusive if you examined the area which you found the bones?
Wait.
So basically, if you went back to where you found them and looked at the environment, do you think you could determine whether or not it would be a viable conclusion that it was 2 deer?
Yeah, so what the thought process there was, they were all in like the radius between like me and Gray, but they could have, the reason why we thought they may have been from 2 was because of, obviously, maybe some animal brought them there to kill them. I'm not sure if studying that area further would give us a better estimate, because we kind of just like saw bones, picked them up, and went back. You right?
In most of your graphs, you included the data of humans or sheep. Why did you think you need to include that?
Yeah, sorry. Basically, I wanted to have the entire picture of this thing up here, and if I were to just cut it off over there, yeah. Yeah, it was a little bit frustrating. I tried to find a website that was just on white-tailed deer, but this is the most, yeah. Emma, sorry.
So, do you have any idea, like, how long the deer had been dead for?
Well, dead long enough for it, obviously everything to have decomposed around it, and the cartilage to have decomposed. So over 8 months, 9 months, but from then on, we don't really know. Dr. Hyde?
So, thank you for your presentation. I'm just wondering, when you came up with the idea that it could possibly lead to, or like more than one set of remains, right? Because it could have been more than one DNA. Did you go back into the trying to maybe check the, making a lot of bones? Did you check and maybe see if there was any obvious duplicates that would suggest, more than one animal?
Yes, so we made sure to check all the tibias, the fibulas, the femurs, the obvious bones that that were more identifiable than the rest. Now the vertebrae, we lined them up, and the ribs, we lined them up. There weren't more than there should be for one deer, so it's still very, very possible that it was just one. But the idea that it was 2 kind of sprung from that, because yeah.
I think we need to go on to our next presentation. Next up is Amanda and Quinn.
Is there a microphone to use?
Hello? Hello? Alright, hi. So, hello, I'm Amanda. I'm Quinn. And when it was earlier in the spring and when the leaves has just begun to come out, we've seen, every day we come into bio class and we see leaves getting bigger and bigger and the trees getting greener and greener. So that brought us the question of how do leaves get bigger during leaf out? And that's our Curiosity Quest project.
We also noticed that this happened for trees that started with flowers, and so that made us really wonder, like, how does that process work? How do trees that first have flowers then grow leaves? And so here are some stages of the leaf out that we took pictures of, specifically in redbud trees, which start with flowers and then grow leaves. So this is the first stage. As you can see, there are many big flowers on this tree and there are no leaves in sight.
And this is the second stage. As you can see, it's an early stage of redbud leaf coming out from the bud scale. The bud scale is essentially an outer protective layer of the leaf bud, which holds the redbud leaf in before leaf out occurs to protect it. And here's just a closer look of the bud scale.
So then the next stages that we see are a tiny folded leaf. You might see that in like arrow B. coming out from that bud scale, and then in C, that folded leaf getting bigger while still folded.
And here, C is just a folded leaf from before, and in D, you can see it really to unfold. And this is just the unfolded leaf after it's fully unfolded. After the leaf has unfolded, it begins to expand until it reaches full development.
So here you can see all the stages. A is our bud scale, then our tiny leaf. That leaf gets bigger, it expands in D, and then in E, we have a fully developed, fully expanded, and fully unfolded red bud leaf. So our question was, how are these leaves getting bigger? So like, what mechanisms are going on inside this leaf to make this leaf get bigger? Yeah.
And just to add on to that a little, our original hypothesis for this was that leaves, well, what we've known before we did our experiment was that leaves, the cells of the leaves divide in order to get bigger. But that's just our hypothesis for now.
Yeah.
And our approach to this is that we took pictures of red bud leaves in different stages of development and under a microscope, and we counted the cells using a program called ImageJ manually, and to see if the cells got bigger or the cells are dividing or whatever is happening.
So this is how we counted the cells. As you can see, each dot marks a cell, and you might notice that some of these dots are dark blue while some of them are light. The dark one's labeled 1 and the light one's labeled 2. That simply shows the leaf vein cells versus the regular leaf cells.
And the leaf vein cells here is actually something we've noticed is that they're bigger than the regular leaf cells, even though we included the leaf vein cells and also the regular leaf cells in our cell count. But it's just good to keep in mind that they're bigger than the regular leaf cells. It doesn't really affect it that much since it's not too much of a difference, but that's just something to keep in mind.
So this is the earliest stage of leaf out. So this is when the leaf is fully closed. And here, this is just a picture under the microscope at 600 times magnification. And this is not really important, but in that picture there are 1.7 million pixels on the screen, and we simply use this pixels idea to see the size of the leaf and how many pixels per leaf per cell the size was.
And this is the second stage, which is a later stage, which this is where the leaf just begins to unfold.
This is a later stage where the leaf is now fully open but not yet fully developed, so it still needs to get bigger and expand. And each of these pictures have the same number of pixels, and each of these pictures are at the same level of magnification under the microscope. So this, any change in cell size that you might see is not because we zoomed in more, it's simply because the cells are actually getting bigger.
And this is the last stage where the leaf is fully developed and fully unfolded.
So here's just a comparison of that first stage of the leaf fully closed versus the fully developed stage. These are both at the same magnification, but as you can see, the cell size is drastically different, and the cells have definitely increased in size.
And if you can see, you can notice the little blue dots. Each blue dot marks a cell. And you can see how close they are in the image on your left-hand side versus how far apart they are on the right. So we counted some roadblocks on the way. So we counted all these images by hand, because when we first thought of this idea, Mr. O'Connell introduced us to a great program called ImageJ, and in there's a thing called automated cell counting, and we tried to make that work. It was really complicated, and it did not work because it was kind of stupid.
It wasn't stupid, but it was not as accurate.
We were smarter.
Manually counted.
Yeah, so we just thought it was more efficient to count them by hand.
Our second roadblock was taking pictures under the microscope. As you can see, terrible picture, how are you supposed to count those cells? This is actually what most of the stuff we looked at under the microscope looked like, and we had to like fiddle a lot with the microscope to see, like to actually find a clear image. And to find this clear image, you have to make very thin, very small specimens of, or like little portions of the leaf to put under the microscope, which is also kind of tricky.
And a third roadblock is that as we look under the microscope, there's actually multiple layers of cells in the leaf. And it's hard to distinguish them and to see which ones to include in our cell count and which ones to not.
So this is some of our data. Basically, this shows the actual size of the leaf. It grows a lot throughout leaf out, and it actually grows exponentially, like by 4 times the amount from the beginning.
And this is our data from our cell count. So basically, we took 4 stages of the leaf. One's fully closed, one's unfolded, one's open, and one's open to fully developed. And we took 6 samples, which basically means 6 images, and we counted each one of them, and we took the average of all of them.
This means that we counted over 5,000 cells manually by hand.
It was kind of driving me crazy by the end of it.
And so this shows the average cell area.
Well, this is just the average cell area, which we got from looking at how many cells there were in an image. If there were less cells in an image compared to if there's less cells in one of the later stages of development compared to the earlier stages, that means the cells are getting bigger. Because the more bigger the cells, the less cells it would have, since it's taking up more space per cell.
Yeah, basically, if the size of the picture is 1.7 million pixels, you can just divide that by the number of cells in that picture, and you'll find how many pixels there are per cell, and that shows the size, which grows exponentially. And so this graph is really important. It shows the percentage of increase from one stage to the next, and we can see that the leaf actually grows more than the cells. So that's kind of an interesting question for us. We're like, what is causing that gap between these 2 lines? This basically shows that something else is also happening to make these leaves get bigger. So the cells are not only getting bigger, but something else is happening. So we're like, what is that?
Which this made us to question that what is this other mechanism that's allowing the cells to, allowing the leaf to get bigger? And one thing we noticed from our area per leaf is that it grows exponentially. And one thing that grows exponentially is cell division. So as we look at it now, we look at the average number of cells per leaf. If there was no cell division going on, the graph would be a straight line going from one side to the other, because there's no more cells adding on, no more cells per leaf because they're not dividing. But actually that's not the case. You see the graph is going up, which suggests that cells are dividing and the number of cells in a leaf are getting more.
Another thing we noticed is that these fully developed leaves seem to have less layers that we can see than the fully closed first stages of the leaf. So that brought us to another idea, but we'll talk about that in just a second.
Yeah, so what does this data suggest?
Our conclusion.
So, of course, as we can see from the image that cells are definitely getting bigger, especially when you compare them side by side.
Cell division is probably occurring because we see more cells, on our graph.
And this came to our surprise, but we think that cells that were overlapping may begin to spread out as the leaf becomes more developed. And that's also another factor.
Yeah, this is another possible explanation for the amount of cells increasing. So what? Why is any of this important at all? Why is leaf out important? Why would the tree have many mechanisms for rapid leaf development? So why would the tree have both cell expansion and cell division, and maybe these layers spreading out all at once.
So this is where we have to talk about photosynthesis and the process of leaf out. So in the beginning of leaf out, as you can see, there's no leaf. So there cannot be any photosynthesis going on. So the tree has to use energy from what is stored from last summer and last fall.
So a fully developed leaf can perform photosynthesis, and photosynthesis only occurs when the leaf is fully developed. So photosynthesis actually creates a lot more energy for the tree than just like using the stored up energy from the summer and the fall. So it's very important for these leaves to like come out quickly and rapidly so that the tree can produce photosynthesis as soon as possible, so that they can have more energy quicker.
So we came to an answer that the quicker a leaf could fully develop, the more advantageous it is. The reason we say the quicker a leaf could fully develop rather than having an earlier leaf out is because when leaf out occurs too early, it would be too cold, so the leaves would die. But when it occurs too late, a lot of energy is used up from what is stored from last summer and last fall, and it would not be good to the tree to do that. So the quicker the leaf can fully develop, when it's the right time, the more advantageous it is, because it can acquire more energy through photosynthesis. Alright, so a cool discovery we made on the way. It's not a very good image, but in the beginning we talked about bud scales, and I originally thought the bud scales was like a teeny tiny leaf, and it was a teeny tiny leaf at the beginning stages of development. But as I put a bud scale under the microscope, we see that the cells are really big, since our discovery is that the earlier a leaf is in the stage of development, the smaller the cells are, but this is not the case. So we figured it couldn't be a leaf because the cells are huge. So it turns out that is a leaf bud. Leaf bud are not leaves, they would not become leaves. And they're just the outer protecting layer of a leaf bud.
So for next time, what we would do differently if we redid this project is basically, one, add more specimens, like collect more data, and then also to get better specimens that are thin enough to actually get good pictures under the microscope, especially for those first stages. It can be hard to get good specimens.
And adding on to this, for the very earlier stages of the leaf development, we couldn't get good images of that because the leaves were closed. That means that the specimens were a lot more thicker. If we could figure out a way to separate the earlier development stages of a redbud leaf, that would give us a lot more data of, especially in the beginning stages of a redbud leaf, and I thought that would be really cool to look at.
So the questions we still have after our research is, do these cells that were in layers actually come out to one surface, or do they just stay in multiple layers? That's still something that's kind of inconclusive. Do all these three processes happen at the same time, and is there a way to measure how overlapping cells might come up to one surface?
And maybe someday we'll figure it out.
Thank you for listening.
Thank you.
Any questions?
Yeah, did you guys ever figure out what type of tree it was? And also, were you like measuring the same tree every time?
Yes, it's a redbud tree, and it's the tree that's right, like, do you know the trash can that's around the circle that's outside of Amos?
Yeah.
Yeah, it's the tree that's right around the trash can.
Okay.
So yeah, that was the tree that we looked at every single time.
Roland?
Well, what does leaf out mean?
Oh yeah, leaf out is basically the leaf coming out. It's the process of a tree going from bare leaves to actual, like, developing leaves. Like, leaf out is just a process of a leaf forming.
Peter?
You said that the leaf vein cells were bigger from the start. Do you notice that these grew at the same rate as the other cells, or did they grow at a different rate?
As far as we know, or as far as I've seen, they do grow at the same rate as the other cells. I think.
Greg?
What was the time span? How long did it take to collect all this?
Well, that's a good question. Since we weren't really looking at time, we were actually looking at different stages. So even now, in one branch, I don't know if I can go back to that slide, it's really early on, but like in one branch, we can't go back to it, it's okay. In one little section of the branch, you can see every different stage of leaf out actually happening. So it the time wasn't it wasn't as important of a part as like the actual development itself. So earlier in time there wouldn't be any of these later development stages, but now if you look at the tree, you can still see those early development stages of branches that are just a bit behind or so.
But it did take a lot of time to count 5,000 cells.
Yeah. Ted? Oh.
So you guys talked about the flowers in the beginning. Did the flowers, or did it work out with the flowers?
Not really. We were mostly looking at the cells and the leaves, but basically what happens is the flowers fall off the tree, and then in their place, you see that bud scale forming and the leaf.
Yeah, if you walk out at Amos and see the same exact tree, there's no more flowers on it, I don't think. It's just green leaves. William?
Did you ever find the mechanism that caused the leaf cells to grow larger?
I mean, well, what we thought of before was it was filling up with water, but I can't testify to how true that statement is, so I'm just gonna say I don't know.
Earlier, you guys talked about how only a fully developed leaf can take on those surfaces. So do you think maybe like the more fully developed tree, like the more fully developed leaves a tree has, the faster this process of cell division and cell expansion happens?
That's actually a really interesting idea. We hadn't thought about that, but I guess that sounds accurate, because once there are some fully developed leaves, some leaves will start performing photosynthesis, and then you'll have more energy, and then the tree will then have more energy to have those other leaves leaf out. So yeah, that's an interesting question.
Zach?
Yeah, so I think when we usually look at trees, we see that the leaves come out first, and then the flowers are formed, and then the tree bears fruit. If the leaves are so important to making more energy, why spend making flowers first before making leaves?
It's a cool question, but I do not know.
Yeah, really cool question. That's something that we hope to figure out one day, but that's actually really interesting.
All right, I think we've really stopped questions there. Thank you for so many good ones. Last presentation now, let's welcome stage.
Hello? Okay. I'm Daisy, and today I'll be talking about my curiosity quest, which is about how does sugar move through plants. So in early February, Mr. O'Connor's biology class all went into the woods, and we tapped a maple tree, if you can remember. And we drove into the tree, and we could see sap flowing out from the tree. And because we know that sap is basically like sugar water that descend from the root to the leaves to supply the leaves, and so that they can grow. So I started to wonder if sap is present in all parts of the tree. Is there a difference between the sweetness level of sap in different parts of the tree? So because we know that the sap goes from the root up to the branches, I predicted that the sugar concentration would be higher the highest in the roots, and then the trunk would be slightly lower, and then the branches have the lowest sugar concentration. And how I would do that is by collecting sap. So I would draw into a tree and then collect sap. And then, from these different locations I marked in the picture. And then, so before measuring the sweetness level, we need to know what actually made sap sweet. The answer is sucrose, because sucrose is the main form of sugar in the sap. So what is a sucrose? It is a disaccharide. Basically, it's like a compound sugar with a glucose and fructose. So now, how can we tell the sweetness of sap? Because we don't have access to a sucrometer, which measures the sucrose level in the sap, all I could find was a glucometer, which was actually what Azir and Well was using in their experiment. Yes. So if I want to use a glucometer to measure the sweetness level, I first need to make sure that it's measurable. Because in the sap, as I mentioned, the main form of sugar is sucrose. So if I directly use a glucometer to measure the sweetness level of sap, I couldn't get a number because it would be under the detectable range. So what I could do is place invertase into the sap. So it would look like this. And so invertase is an enzyme that can break down sucrose. So sucrose would be broken down into glucose and fructose, and then I can use a glucometer to measure the level of glucose in the sap. However, I faced some problems. So if this is the sap that I looked like in February when we tapped the tree in biology class, this was what happened in late March and April when I tapped the tree. So as you can see, sap is not coming out in late March and April. So if I wanted to continue with my investigation on sap, I needed to find another plant in the wood that I can collect sap from. So after talking to Mr. O'Connell, I found grapevines, which look like this. So they basically grow around the trees as a support. And then you can see there's little leaves coming out, and you can see the little grapes, and then they grow into grapes. So you can see from this picture that the structure of a tree and the structure of the grapevine are really different. So I couldn't really carry on my original question to the grapevines. So this is why I had to change my question. So because that I can still collect sap from grapevines in late March and April when there was a rise in temperature, I started to wonder if the soil temperature has an effect on the amount of sap that flows out. So I would expect that the higher the soil temperature, the more sap that can flow out. So my approach would be collecting sap, measuring soil temperature, and then measuring the quantity of sap. So here are 7 different grapevines that I found in the woods, as you can see here. I used a power drill, which you can see here that I taped up a little bit. That's just to make sure that the size of the holes I drill are the same. So I started collecting sap. This time I was able to successfully collect sap. As you can see here, there's a sap droplet coming down from the hole I drilled. So, and then I started measuring soil temperature. I should have done all of this in Celsius, so here we go, I converted all of them. So if I need to put all my data into a graph, it will look something like this. So you can see that I was trying to find a correlation between soil temperature and the quantity of sap that I could collect. But the result ended up like this. So I used a trend line. As you can see there, the blue line. That's just a line that shows the correlation between the two things that I was trying to measure. And if you want to express it numerically, you can use a correlation coefficient, which has, so the highest correlation would be 1, a strong correlation would be like 0.7, a weak one would be like, say, 0.25 to 0.5. So just as an example, you can see that this is a really strong correlation. It has a correlation coefficient of 0.9, and this one would be a weaker one, which has a correlation coefficient of 0.5. Back to my slide, I have a correlation coefficient of 0, which means that there's basically no correlation between the temperature range I measured and the quantity of sap that I was able to collect. But there was a possible confounding variable that I forgot to eliminate, which is the length of the grapevine. Because if you think about this, the longer the grapevine, the stronger the force it would be needed to drive the sap up to the very tip. So like to its leaves and supplying like the leaves to grow. So I would expect that the longer the grapevine, it can also influence the quantity of sap. So I found 2 ways to eliminate this confounding variable. First one is to actually use the same grapevine, but measure it at different soil temperatures. Another one is just to take the length and thickness of each grapevine into consideration. So, solution 1, same grapevine at different temperatures. So I chose this grapevine, as you can see on the left. It's the only one that was still leaking after 4 days that I drove into it. So I was expecting something like this because I was collecting sap from the same hole I drilled. So I was expecting something like it would be really high, the quantity of sap that I first drilled in, and then it slowly goes down until the sap runs out. But instead, I got something like this. So if I put it into a bar graph and compare with the soil temperature, you can see that there was a decrease in soil temperature and there was a decrease in the quantity of sap that I was able to collect. This is not surprising because I was expecting something like the graph on the left, and if it just keeps decreasing, we can't say that it's the soil temperature that made the difference. But what's really interesting is that when the soil temperature increased, it was a really warm day, and then when I went back to collect the sap, the quantity also increased by about twice as much. So that's very interesting because my data then show an increase in soil temperature would possibly lead to an increase in the amount of sap that can flow out. But if I had more time and if I was able to collect more data, then the precision would also increase and it would make my experiment more conclusive. So going back to solution 2, just taking the lens into consideration. Here are just 2 pictures of the grapevine that I was doing my project on. I couldn't come up with a really good way to measure the exact length of the grapevines because, as you can see, that's how the comparison of a person standing under the grapevine versus how massive the grapevine was. So, make it more clear, this is the length of the grapevine on the left, while this one was the one on the right. So you can see that the one on the right is much longer than the one on the left. And it's the same as what I expected, that the one on the left has a lower quantity of sap compared to the right one. But what's really interesting is that the grapevine that I was talking about in solution 1 was actually from the same grapevine as the one that you can see on the right. So they were just 2 roots of the same grapevine. So this triggers some further questions. So I started to wonder, why did the 2 different roots of the same grapevine have a significantly different amount of sap flowing out, and what made the difference? And other than the one that was still leaking after 4 days, all other vines have their bark really wet, but I couldn't really observe like sap actually dripping down from the hole that I drilled after a few days. And I observed that there was this whitish substance that was occurring at the exact spot I drilled a hole. So there's ones that are harder and less transparent, and there's ones that are more like a water droplet and more transparent. I also saw the same thing occurring in other grapevines. So it wasn't only like one grapevine. So this led me to consider why is this occurring? And is this the grapevine's way of trying to recover? And will this whitish substance eventually become harder and harder and become like part of the grapevine? So today I was more or less talking more about the process of my curiosity quest, and there's also questions that were triggered along the way. So yeah, that's it. Questions? Dr. Hart.
Thank you for your presentation. You received lots of questions along the way there. Obviously, you kind of run out of time to present these projects, but what would you, if you had more time, had more resources, what would you like to explore next?
Well, if I had more time to investigate more on this question, I would just observe how like the substance changed over time. Like I was saying here, there was ones that are harder and less transparent, and there's ones that are more like water droplets. So I would observe it and keep like recording the data and see like what it turned out to be, if it actually grew back to the vine, and yeah, like just the time, and see what happens.
The judges are now going to confer, and during school meeting this week, you'll hear which of the presentations the judges deem to be the best. But I think everyone here will agree that all 5 of these presentations were fantastic projects and wonderfully presented. So let's hear a certain